Good morning, everyone. I am Andrea Mazzucato, Associate Professor at the University of Tuscia in the city of Viterbo in Italy. I'm a plant geneticist working mainly in tomato genetics and breeding. This is the first video about the reproductive traits, and it deals with flowering time and the inflorescence structure. As in all plants, the switch from vegetative to reproductive phase is a developmental event triggered at a precise point of the plant life cycle. But the tomato regulates its transition to flowering with endogenous stimuli, being independent from photoperiod or vernalization, differently from other species. On a broad average, the tomato plant produces the first inflorescence after the emission of about 8 true leaves, but the actual time to flowering may largely vary. The time to flowering is the 41st trait of the CPVO protocol, and it is expressed in a 3-classes scale with reference to standard varieties: early, medium, and late. Actually, the protocol gives a detailed quantitative method to record the trait, making reference to the flowering date of the third flower on the second and possibly the third inflorescence of each plant. It is recommended to avoid the first truss as it is influenced by seed vigor. For determinate varieties, the protocol indicates to grow them as the indeterminate ones, and to take the record in the same way. In large phenotypic screenings, it is usually complicated to follow this protocol, as recovering the third flower in each plant may be cumbersome, as well as recording the trait on higher order inflorescence. Thus, in these cases, allowance can be made for the seed vigor effect, and the flowering time is scored as the day of the first open flower in each plant. In this way, the flowering time will refer to the first inflorescence, and it will be expressed as days after sowing, DAS. An even more simplified way to express flowering time is to refer to the transplanting date, provided that plants are transplanted at a uniform developmental stage. Thus, the variability in germination power and seedling development is avoided. In this case, flowering time can be expressed as days after transplanting, and referred to the first or the second inflorescence, as we explained before. An alternative strategy to record the flowering time is to count the number of leaves before the first inflorescence is emitted. This trait is not present in the CPVO protocol. As said, the broader average of leaves emitted before the first inflorescence is 8, but the value can actually range from 6 in some determinate types to 10 or 12 in some flowering mutants. If cotyledons and all leaf nodes are clearly visible, the detection of the trait is very straightforward. However, care should be taken to avoid counting the cotyledon node as a leaf node if cotyledons desiccate, or skipping leaf nodes if basal leaves desiccates, or even skipping the first inflorescence if it precautiously aborts. Finally, we treat here also time to maturity, the 42nd trait of the CPVO protocol, as its detection is parallel to flowering time. The CPVO protocol suggests 5 classes in this case: very early, early, medium, late, and very late. This classification is reached after a quantitative detection as the time to the appearance of the first fully ripe fruit in the second truss, taken on each plant. For the same reason said before, CPVO recommends to avoid the first truss. As for the time to flowering, in large screening, the needing to detect other fruit traits or to sample the fruits may suggest also in this case to refer to the first ripe fruit in the first truss. Once that flowering has been induced, the structure of the inflorescence is the next reproductive trait that we can focus on. The tomato and its wild relatives usually show an inflorescence in the form of a simple cyme that is referred to as uniparous inflorescence. However, in many cultivar genotypes, the inflorescence may branch one or more times, giving rise to multiparous organs. The CPVO protocol ranks the trait into three classes, mainly uniparous, mixed, or mainly multiparous. This classification is quantitatively determined by counting the type of inflorescence on the second and the third truss of at least 10 plants, using 40% and 60% as the borders for each class. Actually, the degree of variation in inflorescence structure may be much higher, and the phenotyper may want to distinguish among different types. The uniparous inflorescence in some mutants, like torosa or single flower truss, can be composed by a very reduced number of flowers, such as 2 or 1. The normal uniparous inflorescence that usually takes the form of a scorpioid cyme may stand more straight, giving rise to a simple cyme. In some cases, finally, the inflorescence can even be longer and more straight, giving rise to a raceme that we call fishbone inflorescence. Also, multiparous inflorescence may present a lot of variation, from a simple bifurcation like in the forked type to more branches like in the irregular type, up to inflorescences that have dozens of branches and hundreds of flowers. These are due to mutations like compound inflorescence and are called compound. Unfortunately, compound inflorescences are usually very unfertile, and less than 10% of the flowers may set fruit. So, if you want to deepen the recording of the inflorescence structure, you may take into consideration more classes. In our phenotyping in Traditom and Harneston projects, we made reference to 5 classes: simple and fishbone for the uniparous one, and forked, irregular, and compound for the multiparous one. This follows an evident increased level of complexity. We assigned a class based on the prevalent inflorescence type estimated by the operator. And in this case, the boundaries between different types should be clearly stated and agreed among operators. If a more precise determination of the degree of compoundness is needed, it is possible to count the number of branches in single inflorescence. This trait is not reported in the official protocol, but we set up to take it in the Harneston screening. Branches can be counted from the second to the fourth inflorescence in a congruous number of plants. This is a labor-demanding approach, but it is actually the most precise way to estimate the degree of branching in multiparous inflorescences. Another inflorescence trait that is not reported in the CPVO protocol, but that we usually consider in our screenings, is the degree of determinacy of the inflorescence. Normally, the inflorescence is determinate, meaning that the inflorescence meristem hands the structure with the last apical flower. In many genotypes, the inflorescence may revert to vegetative development, so becoming a leafy inflorescence. Sometimes the indetermination could even involve the development of a shoot. We refer to this trait as a leafy inflorescence, and use the scale reported in the slide: absent, with leaf, with shoots, or with leaves and shoots. In this case, the trait is intended as the capacity of the phenotype, and a single leaf inflorescence will be sufficient to assign a plant in class 2. Trait scoring should be made in a precise developmental stage, as the trait can change with growth. That is, a leaf in the inflorescence may sometimes develop into a shoot. As an extreme, mutants of the falsiflora gene are emblematically incapable to keep the inflorescence meristem determinate. This gene, that in other species is called leafy, and the name comes from the mutant phenotype, is an essential regulator of the floral transition, and severe mutations like falsiflora on the left in the slide show very indeterminate inflorescence, incapable to develop a flower. Weaker mutations of falsiflora exist, like pistillate on the right. In this case, the flowers can differentiate, although the structure of the inflorescence is still very indeterminate. Okay, this is all for the first reproductive traits, and I wish to thank the local organizer of the course, the Maritz Vegetable Crop Research Institute, the Arneston Project and the Roxy Coast Action for supporting the course. And thanks to all of you for attending the video.